Hello friends, this video on Biotechnology Principles Part 5 is brought to you by examfear.com. No more fear from exam. So now we will understand how is the mix and match of genes done. So by now all we know is that the gene is extracted from one organism and then it is introduced inside another organism. But how this extraction and introduction is actually done. So we will give a brief idea about that in this topic. So first of all, the first step in this would be identify DNA with the desired genes. Now first of all, we should know that which is the desired gene. For example, when we talk about the jellyfish, we say that okay, the gene which is responsible for the glowing nature or for the glow at night. So we are interested in that gene. So that is the desired gene. Now, just knowing which is the desired gene is not enough. You also need to locate that particular gene because on the DNA, you have thousands of genes. So from that, you have to actually identify which gene is responsible for the glow in a jellyfish. Now, once you are able to identify the gene, then what do? You cut the DNA, because now as we all know, DNA is a double-stranded structure which is quite long. Now we are not interested in the entire DNA. We are just interested in that part of the DNA which contain the desired genes. Now by now you all know this is how the DNA looks like, and it is a quite long structure, and as we know that it is so long that it can go up to meters, and that is why it gets coiled up over and again with the histone protein so that it can be accommodated within the nucleus. So we do not want that long DNA strand because we just want to extract a particular gene. So we will cut the DNA based on the specific gene, location of the gene. Let us suppose this is the DNA and we found that the gene is located here. So what do? We will cut the DNA at the specific locations depending upon the location of our desired gene. Now the question is, DNA is not a paper that you will just cut it with a scissor. So how do you cut DNA? So for DNA also we need something which can act as a scissor, which we call as DNA scissors, which can cut DNA at our desired location. Now what are these DNA scissors? So these are again special molecules, and these molecules are the restriction enzymes. So these enzymes act as DNA scissors, that is, they help us to cut DNA at specific locations. So these kind of molecules are called restriction enzymes. Now what happens once these enzymes, there might be several questions in your mind that how an enzyme can cut DNA. So we will understand all those mechanisms a little later. Right now I'm just trying to give you an overview that how the process happens. Now these enzymes will cut the DNA. So you can just imagine them like scissors. Now once the scissor is able to cut the DNA, let us suppose this is the entire strand of DNA and you are just interested in this much portion. So what do? The scissor cuts it from here. So you get a piece of DNA, and this piece of DNA contains the desired gene, which is often known as the gene of interest. So this desired gene is often called as the gene of interest because we are interested in this gene. So this piece of DNA contains the desired gene. Now what do we do with this piece of DNA? Because now our job is to insert this DNA or to introduce it inside the target organism. That is, how do we introduce this DNA inside another target organism? So now the question is, what do we do with this cut piece of DNA? Can we directly insert it into a target organism? Will that help? Well, that really will not help, because if a piece of DNA just enters directly inside the cell of that organism, it will not be able to replicate or multiply itself. Now, if this particular piece of DNA is not able to replicate itself, then our purpose is not solved, because until and unless DNA replication take place, it will not be actually able to transfer its content to the next generation, and that is what we want. We want the desired traits to be present in that organism and also to be present in the coming generations, that is in the progeny. So what do? In order that it replicates, it needs to be integrated with the genome. so that it becomes a part of the chromosome of the target organism. Now, once it is a part of the chromosome of the target organism, then what happens? It will have the ability to replicate, and once it replicates, it will be able to transfer its content to the next generation. So now, how do we ensure that it is integrated with the genome, or how do we ensure that it becomes a part of the chromosome? So for that purpose, we need a vehicle. And what is that vehicle? That vehicle is a vector. So it is a vehicle DNA molecule to transfer the cut piece of DNA into the host organism. So this vehicle DNA molecule, this will actually attach itself with this cut piece DNA, and then it will get introduced inside the host organism, and this vector along with this cut piece DNA together will form a recombinant DNA, which will be able to, or which will have the ability to replicate. So now we will see that how that entire process happens. So please remember that vector, the word vector, you have heard it before also while we were talking about the infectious diseases. You had, if you remember, that mosquitoes are the vectors for the disease malaria. Why? Because they act as carriers. They carry the germ from one person to another person. So they are the carriers. Similarly here also, the vectors are the carriers. So the vehicle DNA molecule, this special DNA molecule which act as a carrier or a vehicle, is known as a vector. So what type of DNA molecule is this? It is generally a plasmid DNA. And what is plasmid DNA? It is a small circular DNA which is capable of replicating independently of the chromosomal DNA. So if you look at any bacterial cell, so inside that cell you will see there is one chromosomal DNA. Other than the chromosomal DNA, you also have a circular DNA which is called plasmid DNA. So let us suppose if this is your bacterial cell, so inside the bacterial cell you have one circular DNA. Other than that, you have one chromosomal DNA. So this circular DNA is the plasmid DNA which acts as the vector. Why it acts as vector? Because it can replicate independently. So it can replicate on its own. So when this cut piece of DNA gets attached to this circular DNA, they form recombinant DNA. And this recombinant DNA has the ability to replicate. And that is our main goal. That is why this plasmid DNA is commonly used in labs for manipulation of genes, because of its self-replicating capability. So the most important feature of plasmid DNA is that it can replicate independently. So it doesn't depend on the chromosome for replication. So it will replicate independently. So this is how the plasmid DNA or the circular DNA looks like. Now, if I want this particular piece of DNA to become a part of this circular DNA, what do we need to do? We again need to cut a portion of this circular DNA so that a space is made available in the circular DNA, and in that space this can come and it can join. So that is how it will happen. So let us see how it goes. So now the next step would be the formation of recombinant DNA. What is the meaning of recombinant? Re means again, like revise, revisit, repeat. So anything which happens again, that is called, that has a re before the word. Combinant is derived from the word combine. So what is happening here? So the piece of DNA, that is the piece of desired DNA which contains the gene of our interest, so that piece of DNA is going to combine with the plasmid DNA or the vector. So this is known as, and therefore the DNA which is formed is called recombinant DNA, because it is formed by combination of a plasmid DNA and a piece of desired DNA or a foreign DNA. So that is often called gene of interest or foreign DNA or desired DNA. So you may call it whatever you wish to. So this is how it happens. So now we have, this is the cut piece DNA. or which contains the desired gene, and this is the plasmid DNA, which is also cut in order to make some space available for this cut piece DNA to attach to it. Now, how do we cut this? Again, the same thing, the restriction enzymes will help in cutting it at specific sites. So therefore, restriction enzyme helps both in the plasmid DNA cutting as well as in the cutting of this desired DNA containing the gene of interest. Now, the cut piece DNA will link with the plasmid DNA to form rDNA. Recombinant DNA is often written as rDNA in short form. So now this is how it will join. So this piece of DNA will get linked to the plasmid DNA. Now how it actually gets connected here. So for the connection purpose, there is an enzyme called DNA ligase which helps in the linkage. So basically DNA ligase acts as a glue which helps this piece of DNA to get stuck in the circular DNA. So here you can see it acts as glue. So what is this glue? These glues are nothing but the DNA ligase enzyme, because it helps in linking the cut piece DNA to the plasmid DNA. And this is how now whatever is formed, this is the recombinant DNA or rDNA, whatever you call it. So now if you see, this recombinant DNA has both the things which we wanted. First thing we wanted was that it should carry the gene of interest. It should carry the desired gene. For example, if you talk about the jellyfish, what was our gene of interest? It was the gene which causes the jellyfish to glow at night. So that gene is present in the recombinant DNA. Secondly, we wanted this DNA to be able to replicate, because without replication, that gene of interest will not get carried forwarded to the next generation. Now, since this has become a part of the plasmid DNA, which is capable of replicating independently, so therefore the recombinant DNA will be able to replicate. So both the purposes are being solved. Now what is left? Now this recombinant DNA needs to be introduced into the target organism. So transfer of rDNA into the host organism. Now we should be sure which is going to be the host organism, so that the rDNA is introduced into the right organism. So now what happens? The rDNA is transferred into the host cell. Now what happens once this DNA enters inside the host cell? Now what do we expect to happen? We want this rDNA to replicate. That is, what is the meaning of replication? That is to create multiple copies of itself. Because when it is able to create multiple copies of itself, only then it will be actively able to participate in the process of reproduction. So rDNA will create multiple copies using the enzyme DNA polymerase of host cell. Now please, if possible, please recollect the process of DNA replication. You remember we saw that DNA polymerase plays the most important role in the process of DNA replication. How? Because DNA polymerase can create new strands after reading the existing strand. So I'll just give you a quick recap. However, I'm not going to explain it in detail. If you want, you can just recap it in one of the previous lessons where I have discussed this. So normally how your DNA looks like. Your DNA is somewhat like this. Now what are these two curved waves? They are nothing but the two strands of DNA. Now both the strands will have nitrogenous bases like this. Now what happens is these two strands separate from each other, and there is an enzyme which helps in the separation. Now when this DNA polymerase comes into picture, what it does, this DNA polymerase will read these sequence. Let us suppose if the sequence here is something like this. So DNA polymerase will read this sequence and it will start creating the complementary sequence here, something like this. So as a result, the new strand will get created. Similarly, this side also the new strand will get created, and that is how you will get one DNA here and you will get another DNA here. So what happened? You started with one DNA and you got two different strands of DNA. So which enzyme helped here? DNA polymerase. So DNA polymerase is the enzyme which helps in creating multiple copies. So here also this recombinant DNA wants to create multiple copies, and it will create it with the help of DNA polymerase, which is present in the host cell. Now this process of replication doesn't start just from anywhere. So in a chromosome there is a specific site where DNA replication starts, and that is known as the origin of replication. So we have discussed all these things in our previous lesson where we have discussed all these. So therefore, for the multiplication of a foreign piece of DNA, it needs to be a part of the chromosome which has the origin of replication. So we need to make sure our main focus is that our DNA should enter inside the host cell and our DNA should actively participate in the process of replication. So for that, you need DNA polymerase which is present in the host cell, and you also need to ensure that our DNA is located in that section of the chromosome which has the origin of replication, because that is the place from where the replication starts. So now once the alien DNA or the foreign DNA is linked with the origin of replication, it starts to multiply, and this process, now when it starts to multiply, what will happen? You have multiple copies of the recombinant DNA. So that organism gets genetically modified because the organism has that gene. So that gene will be shown, or the gene will be expressed in the phenotype of the organism. So the organism is genetically modified, and also its progeny gets genetically modified because that gene is multiplying itself. So a copy of the gene gets transferred to the next offspring, that is the next generation. And similarly again, the process keeps on continuing. So not only the organism gets genetically modified, but the next generations as well gets genetically modified. So this process is known as gene cloning. Why? Because we are creating clones of a desired gene. When you are creating multiple copies, you are actually creating exact clones. However, there might be mistakes by enzyme DNA polymerase, but more or less they are all exact clones. So this process is termed as gene cloning. Now, this is what that happens throughout the process of genetic engineering. So as I said, it sounded very simple that you extract a gene from one organism and introduce into another organism, but there are so many different steps involved in this simple sentence which I said just now. Now also we have discussed it only in brief, only an overview, just to let you understand that how the entire phenomenon take place. Now we will be spending more time to understand each and every step. So we are going to discuss about all the important terms which we have discussed here. Now quite a few things I have underlined here. For example, DNA ligase, host organism, DNA polymerase, restriction enzymes, because they play the critical role in the process of genetic engineering. So now we will spend some time in understanding each of these tools of genetic engineering. Thank you. Please visit www.examfear.com to watch more educational videos with a better experience. Please do not forget to like and subscribe to our YouTube channel for latest updates. Thank you once again.